come to this afternoon session, or third session, which is about the marine microbes. And we will start with a presentation with marine microbes and macroalgae cultivation, and Erik Malta from CT Aqua will present it. So please.
One part of the work carried out in the work package 3, the marine microbiomes, which actually was quite diverse. We did fish, we did microalgae, we did macroalgae, and we did saline crops. So every one of us will talk a little bit about one part of this project. So I will start with explaining why the hell should we grow seaweeds, because it's not as obvious for everybody still. Then I took a more general approach, so I want to go a little bit into why microbiomes are essential for growing seaweeds, what we did in SIMBA and how we contributed to the overall knowledge, and then finally present some more ideas for further research. Okay. So why grow seaweeds? Seaweeds is really, and also microalgae, to be honest, to be fair, is something that's really stimulated at the moment by the European Union. It's considered a very important part of the Green Deal strategy, actually, and it's also mentioned in several of the other strategies. So in the Farm to Fork, in the strategic guidelines for EU aquaculture, and the general conclusion for European Union was that European algae sector, micro and macro, has the potential to become a significant part of the EU blue bioeconomy. So European Union launched a very important algae policy last November it came out. It's communication 592 towards a strong algae sector, I think it's, yeah, towards a strong and sustainable EU algae sector. And there are several initiatives, both private and public, and even combinations. So the Seaweed for Europe is this group of seaweed producers. EU for Algae is an important platform hosted by the EU, but in combination with several partners, discussing all aspects of algae application, cultivation, etc. There's a cost action specifically on sea lettuce, ulva. There's a Safe Seaweed. So there's many initiatives, both private and public, on seaweeds. And why is this so important? There's a large demand actually for algae and algae-based products, and then you also have to think of phycocolloids as agar, carrageenans, alginates that are very important ingredients of many products, not only food and feed, but also many other products. And the EU expects a demand in algae to increase by 2030 of 3,000%. I'm not sure how realistic that is, but they, let's say they expect a very important increase in the demand. And it's based on the fact that Europe is importing a lot of algae biomass, worth of $500 million. Demands for microalgae as chlorella and spirulina are growing by 5% or more per year. And also there's a lot of interest. There's, of course, we have the growing world population. We are going to have to rely on seafoods to just for our protein, for other foods stuff. So yeah, that's where this figure is based on. And there's many applications already known and upcoming. So we know foods, direct food, but also more processed food is coming from seaweeds. For animal feed, we have seen several examples of pilot scale applications for introducing micro and macroalgae in animal feeds. A lot of demand for functional biomolecules for all kinds of applications, pharmaceutical, cosmeceutical. Quite new is the methane emission reducing compounds. So you introduce it in animal feed for cattle and you can reduce their methane emission. It's a bit controversial, I think. Bioplastics is an upcoming sector. There are several companies now working, producing bioplastics from seaweed. That demand for biomass is obviously huge. And then there are the more controversial claims for biofuels and carbon sequestration. I won't go into that, but okay, there are still demand from that as well. So, and then we have to contrast our own seaweed production. 2016, it's a bit higher now, but it's still less than 300 kilotons. which is slightly less than 1% of the world production. So that's where we are. 95% of all algae, and specifically seaweeds, is produced in Asia, mainly by Indonesia, by China, Indonesia second. Then there's Japan, Philippines, etc. Then there are some African countries, South America, mainly Chile, and then in the end there's the US and us. We're really completely insignificant production. And also remarkable is that 98% of our production comes from wild harvest. While on a global scale, it's the other way around. Only 3% comes from wild harvest and the majority from cultivation. And we are seeing the effects. There's already overexploitation of wild communities. So the answer, the new biomass in Europe should come from sustainable mass cultivation and no more from wild harvest. Okay, then we switch to the microbiome part. So why is microbiome so important for seaweeds? And actually, you know already for a quite long time, since the 70s more or less, that bacteria are crucial for seaweed morphogenesis. So this is an example from my own pet species, Ulva sea lettuce, which is, you can have a tubular or foliose morphology. This is a folio species, and if you grow it normally in an open system, it gets this nice blades. But if you grow it sterile, you'll get this very strange morpholysis, this urchin-like morphology, or just a weird callus type of thing. And actually, that is known since the 70s, but the mechanism and later even the species involved, it was only unraveled quite recently. So there was a molecule identified by Matsuo et al. In 2005, what they called tallocine, and that's the molecule responsible for the morphogenesis, and it comes from the bacteria living on the ova and the ova holobionts. So, and actually now in the last decade, more or less, Thomas Wiegard's, especially Thomas Wiegard's group, did a lot of work on the bacteria involved in that, and he created some kind of minimal microbiomes for a tubular ova species, Mutabilis. Well, actually it can have both morphologies, but he mainly works with the tubular. So with 2 or 3 species of bacteria, you can get a nice, healthy, and morphologically right ova. So that's the basis of these microbiomes. Okay. We also have seen some works that found a growth stimulating effect of certain bacterial species on algae. So there's one from India, Gasselaria. Gasselaria is an important agar-producing species. And the graphs on the right are from work also with ulva. Ulva is kind of the Arabidopsis of the seaweed work. So it's very easy to grow and easy to experiment with. So, and there they added different species of bacteria to, I think, a sterile control, and they found growth stimulating effects. So they really experimented also with minimum microbiomes. And more work has been done also of molecules exuded by bacteria growing on seaweeds and used by the seaweeds. The majority of the work has been carried out with ova, but some other species were also used. Okay. Then a very important part is also the role bacteria play in spore formation, spore release, and spore settlement. And this is crucial if you for seaweed cultivation. If you are starting with seaweed hatcheries, which is actually the way that if you go for mass cultivation, you will end up with companies growing seaweeds, companies producing lines, so hatchery companies, and processing companies. So that's a very important aspect of the cultivation cycle. And again, most of the work has been done with ulva. You see Enteromorpha, but that's the old name of ulva. And only a few other species. So, okay, so what we first identified a bit, the data caps and the challenges that existed when we started writing the SIMBA proposal. And actually a lot of has happened in 5 years in this field. So, but what we saw then that most studies that far were fairly descriptive. So there were descriptions of microbiomes of ulva, mainly on ulva actually, in seaweed populations, but they mainly focused on differences or variation between sites in a certain region or between populations, but it did not take into account temporal variation or other factors. Experimental work mainly focused on morphogenesis and sporulation and not so much on growth. There was very little known on the population dynamics, as I said, the temporal variation, and really nothing about growing seaweeds under different cultivation conditions and what would happen with the microbiome population, community, sorry. And even less how these might affect growth or chemical composition. A limited number of seaweeds has been studied. Well, we did not change that in SIMBA. We continued working with ulva. Yeah, that's something for the future. It will be very necessary. And actually also the existence and the specificity of these algae-bound communities needed further confirmation. Still not really clear how different they were from their environment. Okay, so objectives for the work in SIMBA, our model species was ulva, specifically ulva anoe, a foliose form that's doing quite well in warmer waters. So I'm from the south of Spain, so yeah, that's the one I work with mainly. And for a starter, we wanted to analyze the microbiomes of the seaweeds growing in different cultivation conditions, where we had an increasing amount of control about the growth conditions. So we had a floating cage system where we inserted seaweed biomass in a natural environment. Then we had outdoor tanks with controlled and more or less treated seawater where we controlled nutrients, and we had photobioreactors where we controlled most important variables except for pH. So we did a monthly monitoring of the microbiome growing on these algae. We measured growth, biochemical composition, etc. Okay, I'm now going to focus mainly on the microbiome data. So in these figures, what you will see, the green bar is microbiome on the seaweed, the blue bar is microbiome in the surrounding water, and EP stands for earth and pond. So in the natural environment, OT is outdoor tanks, and PBR is photobioreactors. What we saw, first thing that diversity, species richness of the community growing on the algae under natural conditions was quite different from the waterborne community. So we saw here especially both Shannon diversity and species richness in the EP cultures was much higher on the algae than in the water. That also has to do with the fact that if you the algae from the water from natural waters are not really clean. So there's all kinds of things on them, also animals. So I think, although you rinse the algae before taking your sample, but I think you still have some of that microbiome as well. It's part of the community actually, because maybe that's the reason that we did not find these differences so much in experimental communities. However, if you look at the composition, it's here in this, it's really clear. So on the right side are all the water-borne populations, communities, and the left side are the bacteria growing on the algae, and they're completely different worlds. They're completely separated, and they really have nothing to do with each other, despite the fact that they're in continuous contact with the water. So I found that quite amazing, actually. Then we also see that if we focus a bit on the communities growing on the algae, we see that there were some differences, especially with the seaweeds growing in the natural environment. They were quite separated from the other two, and there was more overlap in the two experimental communities. So we see that the different cultivation conditions, they lead to different microbial communities, which might be expected. But it was nice to confirm. But the main thing is here that we confirm that seaweeds, they really have their own community, which is very different from the surrounding water. This is a bit of a complicated figure. It mainly is a kind of a time series of the main, you're not able to read, I understand, the groups of bacteria, but that's not really relevant. The most important thing here is that you see that, so this is the time series, and the community changes actually quite a lot over time, even in the photobioreactors, where they're growing all year round at the same temperature, same nutrients, same light. Even there you see that the community is quite dynamic, and even more so in the water. Water, of course, comes from the outside, but it is treated, it's cleaned, it receives UVC treatment, which may not have been that effective given the diversity and amount of bacteria we still found on them. But yeah, it's important to see that the communities are quite changing all the time, and something that we might actually should model. But I think the behavior is quite chaotic. That's probably, there's a constant competition between species, microbial species, and that might give rise to these dynamics. But it will also good for us, because we can now have a list of all these species occurring there and try to compare what we know from the literature and see what species might actively be beneficial. So that's something we are still working on, actually. And I made a list of the most common bacterial species. That's the one on the left. I try to highlight which ones or species or families or even phyla are known as having a positive effect or having a negative effect, or actually might have both. A negative effect is not really certain. It's a negative effect that are bacteria that are capable of degrading ULVAN, which is the main membrane polysaccharide. But it does not necessarily mean that they attack living algae. They may just be able of degrading already dead cells or something. So, and what we see that of these lists of described species, we did not find them, or at least they were not very abundant in the species we found. So we have a completely different set of species. And that offers the possibility to check, to focus in on these, especially these families where we know that they are candidates for having beneficial effects, and to see if they are maybe better adapted for warm water, because it has to be mentioned that most of these names come from seaweeds from colder waters. So maybe there are specific bacteria families or species genera for more warmer waters. And also for the negative, the algaecidal bacteria, well, Vibrio is of course always bad, whether you're algae, human, fish, doesn't matter. But some of the others might actually, in the end, be beneficial by cleaning up old cell material and giving the other cells more light and space. So that's still a bit of a line to advance. Okay, so to summarize main results, the community we found on the ULVA was very different from that from the surrounding water. Both the ULVA and water microbiome community are not fixed in time, not even under constant cultivation conditions. Little is known still about the actual interaction between the different bacterial species on the one hand and with the seaweed on the other hand, because many species are involved and co-occur. We found a few known negative bacteria, but actually no positive. So there's maybe much unknown potential we have to explore. And we need to still do some more detailed data analysis. And then for future areas for future research, so we need to work on a better understanding of the biochemical interaction between bacteria and algae. Obviously also, so for microalgae, you might be able to get increased B12 content in seaweeds with the right bacteria, so that might be an interesting line. Identification and testing of more beneficial bacteria to construct alternative minimum microbiomes that may have a growth stimulating effect. Compare effect of microbiomes of spore formation release and settlement. They are crucial for future hatchery companies. And there may be very nice applications for engineering the holobionts. And this is an example from a Spanish group that manipulated the ova holobionts, introducing a Phaeobacter bacteria. Then they used the ova in a recirculating aquaculture system. And the Phaeobacter had a little stimulating effect on the ova, but more importantly, it had a great antibacterial effect against fish pathogens. So this is a whole new avenue of potential application. So thank you very much.
Okay, there is one urgent question. During the Erik will take the...
Fascinating. I just have one question. Did you consider to include also the protists because they are in the microbiome and they are fantastic? And Labyrintula is one of the genera. And so eukaryotes, please include eukaryotes also, not just prokaryotes.
Yeah, that would of course be also for another project that has to be, we are a bit limited with funding. But yeah, no, of course, also both for positive and negative effects, because there are some protists that are quite nasty actually, and known,
Very good enzyme producers.
Thank you very much.